Second session consists of 3 virtual speakers, speakers from different parts of the world who are dialing in to be part of this symposium. At this time, I'd like to welcome Dr. Peter Axerio-Cilies. Peter is also one of our ACD Fellowship awardees. He has a PhD in neurology and experimental medicine. And he works as a postdoctoral fellow at the University of British Columbia in the Department of Medicine and Psychiatry. He works very closely with Dr. Sylvia Stockler, and together in collaboration with Dr. Jonathan Schlabach and Chuck, whom you heard from earlier. Today, he will be presenting on some of the work that he has done with patient mutation-bearing fibroblasts and for the functional characterization and drug screening efforts in them.
Thank you for the introduction, Sangeeta. I'd like to apologize to everyone that I wasn't there in person. So yes, my name is Peter Axerio-Cilies. I work at the BC Children's Hospital, and I work with Dr. Sylvia Stockler. My project is similar to Charles's project, where we're screening for drugs that could rescue the creatine transporter deficiency. So our strategies are to find expression correctors. So in cases where the transporter is trapped in the ER, whether we could find drugs that can promote increased trafficking to the cell surface. And option 2 is where you might have a normal expression or maybe a sufficient expression at the cell surface, but is dysfunctional. So the mutation caused a inability to influx creatine. So we could possibly find modulators or drugs that can modulate the transporter to make it functional. And in combination 3 is where possibly when it's trapped in this ER and it's dysfunctional, and by promoting it to the cell surface, it's still dysfunctional. And in that case, combining 1 and 2 together could make it more active or active. So the long-term project aim for our project is to identify an FDA-approved drug or natural product that could either enhance the function at the cell surface or increase the cell surface expression of SLC6A8. And ideally, we want to be able to take it orally, safely, and daily routine. It has a good safety profile and no reported human toxicity. And the therapeutic dosage tested in vitro in our cell is achieved in the brain. So it's useless to have a drug that's, the concentration used in vitro cannot actually get into the brain. And the drug has to be able to penetrate the BBB, so this barrier in the brain. And ideally, the drug itself needs to have a concentration that's low and strong enough. So we want a low micromolar and nanomolar affinity for the transporter. So there are different stages that we're implementing. Stage 1 is diagnosis. So we're classifying in the presence of creatine, how many variants are partially dysfunctional, completely dysfunctional, maybe it's benign, or it could either be, we haven't seen it yet, but hyperfunctional, where it's overactivated. Stage 2, we are creating models, which is what Charles has been doing, we're creating our own models with this new homology model with other similar proteins that have been already characterized, and this new technology called AlphaFold. And we're using in silico screening 20 million drugs to screen for different sites on the transporter. Then we're doing is testing these drugs in our in vitro drug screening. So we're using hex cells and different, which I'll discuss later on, different ways to get an output for these drugs. Then we're going to evaluate them, the compounds themselves, do different doses. And then stage 3 is an iterative process where initially you just find drugs that just work a little bit and then try to optimize them further to get them as close to 100% as possible. Then stage 4 is sort of the last step where we're kind of testing in fibroblasts and BBB penetration and ideally animal studies. So as mentioned already before, there's a lot of variants. So we've made them in the lab and tried to characterize them as much as possible. So there's about 170 in different databases, already in the LOVD database and the Baylor database. And we've picked them based on occurrences. And obviously, the ACD helped us choose the most important ones or most frequent ones. So the first step was to actually create these models. So the models were tested, were made by different strategies. So using homology modeling with other transporters, as mentioned before, the last talk, and also using this new technology that came out with AlphaFold that was able to, it was a huge event, and we used that to create both the inward-facing and the outward-facing for all these type of mutations. And we use these models to screen a lot of drugs. So we thought about doing both the FDA-approved library and the natural product library, but also other drugs. And the idea here is to find a drug that is potent, strong enough to bind to SLC6A8, and then map it into an FDA-approved drug. So once we know what binds well, then we can find drugs from the FDA-approved library that does the same thing. So there are different sites that can be targeted. You can target the creatine binding site. You can target other sites localized in different areas of the protein. So we tried to take a broad approach and try to look at different sites and screen drugs on different sites. But we started with the creatine binding site as that was the most obvious, and then try to find drugs that will be strong enough to bind to the transporter. So this is a small movie just to sort of show how the screening is done. Obviously, this is done in the background. I'm not actually looking at it, but each one of these drugs gets ranked based on how well it binds to the transporter. And then we can look at them manually to see whether it all makes sense and which ones would be purchased and then tested. So when the testing, we use different methodologies. Here we use electrophysiology. So electrophysiology is a way to see the difference in membrane potential. So sodium and creatine get through the transporter, and you're able to actually see the change in current. So it's just like a battery. And you can actually get an output. And then if you see a sort of what I see in the bottom here, if you see an increase or a decrease in this current, you know that something's actually getting through. So here in this case, we know that adding creatine to the cells, we can actually see a difference in current. Then we use mass spectrometry and creatine uptake assays. And for surface expression, we use biotinylation assays just to see how much protein is actually at the cell surface. So initially, we try to do as many mutations as possible, as described in the databases, and try to see in the presence of creatine, how many of them are actually pathological. So here we see some residual creatine activity for some of them. Most of them are null or kind of dysfunctional. And so we're trying to test as many mutations as possible. And the reason here is it will help doctors, certainly this information can be helpful, and also get a baseline of how this mutation behaves in the presence of creatine. So initially screened through wild type, and we've seen in this particular case, we're trying to find drugs that could allosterically modulate the transporter further. And the idea here is that the drugs that can do this in wild type normal can be then tested with the mutations. So we saw around 6 hits with 40 to 100% increase in transporter activity. Then we took these 6 drugs and we tested them in different mutations. So this is what I've shown before. Here we showed that 4 out of the 6 actually showed some partial 20% restoration. And again, here in a different mutation, we saw an increase in 20% as well. They're different in each mutation, which is expected. So every mutation has its own behavior with different drugs. So really all drugs have to be tested in different mutations to be able to know how strong they are. And then what we did is we actually did an enrichment. So enrichment means that we've taken 6 other drugs and try to find some common features. What are the common features of these 6 drugs that is common, that is likely the reason why it binds to that particular pocket? So we rescreened the 20 million drugs, which includes the FDA-approved natural library, and we try to find similar drugs that have that core, a common core. And we use this core to kind of enrich the screening. And when we did that, we found that we were able to find some of these drugs that had much better transporter function, approximately 50% restoration. And we've tested many of these analogs for each individual of the initial 6 hits. And we found that we were able to get an improvement. So idea here is to find other analogs within the same class. And the idea is to find the best interactions and the best drugs. And then we can map these drugs to the FDA-approved library to find something that would work and could be more applicable for testing in humans. During this, all the results so far have been in the presence of creatine and tested on the day of the experiment. So as soon as I add the drug, I saw that increase. In this case, we actually incubated some of these drugs, a different drug, 24 hours before, and we were able to find one expression corrector that showed an increase in activity for all these different mutations. Some of them are still dysfunctional. It may be because of other reasons, but at least we were able to see 4 out of the 10 here that showed a partial increase. So in summary, we were able to test 24 variants. 6 of them showed residual effects. Then of the initial 6 hits that were found initially, we were able to improve it by 50% of the 4 out of the 6 drugs. And we're going to continue on going down this path to try and find the most of this class to see whether we can find more potent drugs. And then we found that non-functional mutations can be rescued in some cases, even if they're entirely dysfunctional. So next steps, we're going to further optimize the lead drugs and then map them out into the FDA-approved library and natural product library. And then obviously test these drugs in the fibroblasts, which we're currently doing using mass spec. And we obtained these from the Coral Institute. It's really important to have really these kind of fibroblasts because they're the closest to a human as we can get. And so there's not many out there. There's only a few that are up there right now, so it'll be useful. And then expanding the variant diagnosis panel for all known variants would be beneficial for, like I said, doctors and physicians. So I'd like to thank for the ACD for the funding, and I'd like to thank Laura and Geetha particularly. And I'd like to thank for the collaboration with Jonathan and Charles, which has been a huge help in finding drugs for this disease. Thank you very much. And thank you for listening.
Thank you so much, Peter. That was a wonderful presentation, and it's really exciting to see how far we've come in being able to gain more insights into patient fibroblasts and patient mutations. Thank you for that. Please stay on until the Q&A panel. At this time, I'd like to introduce our next speaker, Dr. Maurizio Balestrino. Dr. Balestrino joins us from Italy. He is a researcher and a clinical neurologist affiliated with the University of Genova in Italy. His work aims at bridging gap between experimental research and clinical therapies. In January of 2022, Dr. Balestrino and his group published a paper on the delivery of guanidinoacetate-like compounds to neural tissues and their relevance to creatine transporter deficiency. That is the focus of his talk today. Dr. Balestrino, can you hear us?
Yes. As you said, I'm working at the University of Genova, Italy. My name is Maurizio Balestrino, and this is the list of the people who collaborated with me in the research. Now, the background of my research is that we have been searching, like other people in the past years, we have been searching for compounds that may cross the blood-brain barrier and the cell plasma membrane independently from the creatine transporter to try to overcome creatine transporter deficiency. We and all others like us have focused on creatine-derived molecules, but as you certainly know, it has been suggested, mainly by the group of Olivier Bressan in Switzerland, that normally neurons do not uptake creatine as such, as you see in this cartoon. Rather, they take up guanidinoacetic acid, which they receive from the oligodendrocytes. So the theory goes that oligodendrocytes take up, actually they produce, guanidinoacetic acid, they release it, guanidinoacetic acid is taken up by neurons and then inside neurons transferred into creatine. This can no longer happen in creatine transporter deficiency because the same transporter that carries creatine also carries guanidinoacetic acid, then the whole process is blocked. With reason that we found a ProDrug that is based not on creatine but on guanidinoacetic acid, then this would probably be a rather smarter way of increasing, at least a more physiological way of increasing creatine in creatine deficient neurons. For this reason, we designed several drugs and we chose this diacetyl guanidine acetic ethyl ester. The abbreviation is diacetyl GAAE, and it is a lipophilic prodrug based on guanidinoacetic acid that we would like to test to see if it can correct creatine transporter deficiency, at least in vitro. Now, one main problem with this approach is that guanidinoacetic acid is thought to be epileptogenic. In fact, we know that in GAMP deficiency, this substance, guanidinoacetic acid, increases and is thought to be the reason for the epilepsy fits that these patients show. Here you see a cartoon taken from the literature, and I'm not going through it, but we all know that guanidinoacetic acid is thought to be epileptogenic. So is it wise to try to treat patients with this drug, or at least it's a ProDrug? Well, the fact is the evidence for the actual fact that GAA is epileptogenic is not strong. Not only that, but GAA has been used in a number of research papers as a dietary supplement in humans with no adverse events. So, in my opinion, it is not so specific, so so obvious that guanidinoacetic acid is actually epileptogenic, at least in the traditional sense of the word. Part of the explanation may be that different concentrations of GAA were used in different researches. Like now you can see here that in the cerebrospinal fluid of normal subject, guanidinoacetic acid is in 0.0101 micromolar range. In GAMT deficient patients, it goes up to about 10 micromolar range. But in most experiments investigating its toxicity, it was tested at least at 10 micromolar and up to 10 millimolar range. So I think one main problem is what concentration of guanidinoacetic acid we are talking about. To start our research, we studied in brain slices in vitro a concentration of GAA which is identical to that is observed in GAMT deficient patients, namely 10 micromolar. As you see, it actually did not increase the amplitude of the postsynaptic population spike. That is, it did not change synaptic transmission in our slices, especially it did not change it in an epileptogenic way. Epileptogenic compounds do decrease the amplitude of postsynaptic population spike. You will see this amplitude increased 200, 300% and more, and they will show a burst. But at the very least, they would increase the amplitude of a postsynaptic population spike, which you do not see here. If we increase amplitude about 100 times to the very unphysiological concentration of 1 millimolar or 2 millimolar, again, there is no change in amplitude of population spike. If we go even higher to 4 millimolar, we see actually a decrease in population spike, which is reversible upon washout. A decrease in population spike is a hallmark of toxic compounds, but not of epileptogenic compounds. These are additional experiments, preliminary results, additional experiments we are carrying out now. This is in neuronal cultures on microrectal arrays. You see cortical neurons on the left, hippocampal neurons on the right. You see this is on the y-axis the mean firing rate of the neurons. 0 is baseline. You see when we add 1 micromolar GAA, which is the normal concentration in normal people, you see very little change. When you go up to 10 micromolar, which is the concentration in GAMT deficient patients, you see a decrease in firing in cortical neurons, no change in hippocampal neurons. When you go even higher, you find a decrease in firing rate both in cortical neurons and in hippocampal neurons. So again, even in this model, from these preliminary results, at least it does not look like there is an epileptogenic effect. A word of caution comes from this paper by the group of Maurizio, which showed that upon chronic administration, guanidinoacetic acid at the 10 micromolar concentration, which is that of GAMP deficiency, causes excessive neuronal sprouting. And this excessive neuronal sprouting may indeed be epileptogenic, but this is for chronic administration in the developing brain at a rather high concentration of 10 micromolar. So this is the landscape about what we think that we can say on the supposed epileptogenicity of guanidinoacetic acid. Then we go test our ProDrug, diacetyl-GAA, which is rather safe at a 100 micromolar concentration. We are not showing it here, but it is also very stable in water, which is important. And in this slide, we show that diacetyl guanidinoacetic acid, ethyl diacetyl-GAA, is significantly effective in countering brain slices vitro the harmful effect of creatine transporter block. The bar at left shows that upon blocking creatine transporter with GPA, viable slices decreased to about 30% of the total. Upon treating them with our ProDrug, this percent of viability increases to about 50%, which is about twice as much. And this is, as you see, statistically significant. Now, the surprising thing is that our ProDrug did not increase creatine nor phosphocreatine in neurons. Upon, of course, it did so when the creatine transporter was functional, but it did no longer so when the creatine transporter was blocked. So we are rather puzzled by the fact that we do have an effect in the preceding slides, but we do not have increased phosphocreatine. We think that the answer to this puzzling finding comes from this reassuring finding, which was found by the group of Isbrand several years ago in the muscle of GAMP deficient patients. There is no creatine, but the muscle still is able to maintain its function because guanidinoacetic acid, which is accumulated in these patients, can be phosphorylated, and this GAA, phospho-GAA system sets up a alternative system which can vicariate the function of the missing creatine phosphocreatine system. This is the identical way how cyclocreatine works. Cyclocreatine does not increase brain creatine. Instead, it sets up a cyclocreatine phosphocyclocreatine system, which again vicariates the missing creatine phosphocreatine system. So our conclusion is that a ProDrug of guanidinoacetic acid may be a safe and effective way of countering the harmful effects of blocking the creatine transporter in brain tissue. However, we acknowledge that further research is needed to fully understand the translational possibility of this pathway. Thank you for your attention.
Thank you, everyone. I'd like to invite our virtual speakers to please turn on their cameras so that we may proceed to the Q&A session. And I have with me Dr. Jenny Goldstein, who will be moderating this session. And I will let Jenny introduce herself.
Thanks so much, Sangeeta. It's great to see everybody here and just a wonderful experience to meet everybody and to hear these great talks this morning. So thank you so much to our speakers. These have been really interesting talks and really just shows the huge amount of work that's going in to this field. So we'd certainly like to hear questions from all of you. I'm sure you have a lot of questions. But just wanted to start out with Dr. Ferrada. Thank you so much for that wonderful talk, especially looking at variants of unknown significance in the creatine transporter, because these are variants that we just don't know, are they causing the condition or not, and really helpful to understand the mechanisms of pathogenicity. So when you think about this, when the protein is made, sometimes that protein is just unstable and it degrades, and then of course it can't do its job. Other times the protein is made, it gets into the endoplasmic reticulum and just gets stuck there, so it's not able to go out to the plasma membrane where it does its job. And then with other variants, the protein can get to the plasma membrane, but it's just non-functional. So this was really, really great work looking at these different mechanisms with different variants of uncertain significance, and which certainly will improve our understanding of these different variants. So one question that I had was, can the methods of characterization determine whether loss of function is caused by this mislocalization, lack of creatine binding, or both? Is that something that you can address, Dr. Ferrada?
Hello, thank you for your question. Apologies, I cannot make my camera work at this point. There is some error message.
No,
I hope you can hear
and
well.
just for all the panelists as well as our audience here, apologies, we have some technical difficulties. So we're gonna go ahead and do this without camera.
Okay. Thanks for the question. Yeah, this is exactly one of the points that we wanted to make, and we wanted to select these techniques we use because we wanted to differentiate between these two important mechanisms. So there is in the field some techniques, computational techniques that allows to classify variants that are highly unstable, but it only works when the signal, right, is very strong. And this is the case for the example of 8 different variants that I show during the presentation. And usually it works well when the variants are hidden or have low surface accessibility. So that means are usually hidden inside the structure. Or the mutations produces strong changes in the volume of the residue. And so that usually is the mechanistic cause, right? And interestingly, in our case, that coincides very well with the fact that they become unstable and they accumulate, right, most of them in the ER as the current mechanisms show. So that was reassuring for us. But still, these methods need a lot to improvement. They do not perform 100% accurate. And so in order to address that, we're using several methods that I didn't have time to describe, but we're confident that it can get to be reproducible, and we're glad to see that in the experiments too.
Great, thank you. And how about variants for future study and maybe considering clinical testing and helping to interpret those variants, whether they are causing the condition or not? So thinking about databases like ClinVar or also LOVD, which was mentioned in another talk. Are you thinking about continuing to classify additional variants?
Yes, we are. So this is just the first batch of analysis that we wanted to make, in part because we're starting the project and we wanted to validate some of the analysis. And we're looking forward to assess some of the variants that I described in using these other methods that have the advantage of being what is called in the field unsupervised, meaning that there is no human being saying in advance, a priori saying this should be a disease, this should be benign, but rather classifying them according to either evolutionary information or groups that are functionally co-evolving. And we're planning to continue our work with those two. Yes.
That sounds terrific, and certainly helpful to potentially have some of that information ahead of time before these variants are actually found in patients. So thank you so much for all of your work. That was wonderful.
Thank you.
Thank you. And so the next talk from Dr. Axerio-Cilies. So again, this was a fantastic talk looking at functional activity of SLC 68 variants and looking at different compounds that can affect the activity. So just curious, how long did it take you to test the 100 compounds against the 24 selected variants?
So it would be, so we tested it it's like probably, I think, a month. And we do test it at different concentrations. So we try to do a low dose and a high dose just to make sure. But yeah, it's about a month.
Wow, so that's a really fast throughput, which is really fantastic. What about, so it sounds like this approach you were using was looking mainly at missense variants, is that correct? Or do you have an approach for null, like frame shift and nonsense also?
Yeah, frame shift, so deletions, absolutely. So we're definitely looking into that. And in terms of truncations, the only thing is that we are looking at it in terms of testing, but the probability for these drugs to work in case where it's highly truncated or like the large deletions, that would be probably low simply because there's not enough protein that's being anchored at the surface, and probably it's not even likely not made or not made properly. So we do test it, but it's low probability for that.
Jenny, can I ask a question?
Please go ahead.
So, Peter, in your talk, you showed that there are at least some drugs that are able to rescue the function of some of these mutant proteins, almost up to 50% that of normal. Can you comment on whether that kind of rescue is going to be meaningful for clinical aspects of the disorder? So in other words, is it enough?
Yeah, so that's a good question. I think it's hard for me to say whether it's enough. I think the most important thing would be to know how much of the drug would actually get into the brain and actually be local to in the brain. So in terms of whether we actually get 50% in the brain, I don't know, but for sure the idea here would be try to get as high as possible so that even if small portion of that actually gets in the brain, it would still work. So it's hard to answer that question unless to actually test it.
No, thank you. I appreciate that perspective. I guess a follow-up question would be, because we don't know that level of rescue is actually going to be sufficient, are there other models, such as the known mouse models, that you might consider testing these in? And I ask this question specifically because the available mouse models do not show a complete overlap and phenotype with what is seen in individuals. So given that, how important would you consider testing in that models and the results from something like that?
Yeah, no, absolutely. I think having a good model that is consistent with what we see in patients is really key, because in cells you can only do so much and say, yes, we see an effect, it's great that we see that say 50%, but it's really important to actually get a good model, animal model, to say, hey, these drugs are working in the animal model, and then that is consistent with what we see in a human. I think that's really key. I think for everyone to be successful, I think we need those models to be consistent with clinical output. Absolutely.
Sorry, Peter, one more question.
Yeah.
There are cells from patients expressing those variants. Have you tried your system in fibroblasts as well, or your electrophysiological system is better suited just for the HEC293?
Correct. So good question. So we're testing this currently. So we actually are growing fibroblasts right now in our lab. And I agree with you that mass spec is much better than EFIS to characterize in fibroblasts. And the reason for that is because the morphology of the cell of fibroblasts are actually, the surface is stronger. So the patch, it doesn't work well. So we do have a mass spec. So we're going to test some of these drugs. First, the controls, the creatine, and then some other positive controls on these fibroblasts. And also we have gotten wild type fibroblasts as a control as well. So we're going to do this in the next month, this month and next month.
Great. This question is for Dr. Balestrino. I was told you can't see us, so this is Steve Baker from University of Utah. So my question is, do you feel like the physiological substrate for, excuse me, neuron creatine generation is GAA, or do you feel like it's creatine that's taken up from the CSF?
I cannot hear you anymore.
Oh, okay. So can you hear me now?
Now, yes. Hi.
Okay. So you mentioned that neurons take up GAA from the CSF and then generate creatine. The concentration of creatine in the CSF is roughly like 50 micromolar. And you mentioned that the concentration for GAA is something on the order of 1 micromolar. And I was wondering if you feel like neurons mostly take up GAA and generate creatine, or do they take up creatine directly from the CSF?
Well, this is a very interesting question. The neurons do have the creatine transporter. However, they seem to have it at a lower extent than astrocytes or oligodendrocytes in particular. So to start my reasoning, I just started from the theory of Balestrino, which is what I showed. And that theory goes that guanidinoacetic acid is what the neurons actually uptake to make creatine. This does not mean, in my opinion, does not at all mean that the neurons cannot uptake creatine. But this means that this is not their preferred way of function. Also, the concentration in the CSF does not necessarily reflect the concentration in the interstitial space. So there may be different about that. But I don't know if I answered your question in a satisfactory way, but I'm afraid that this is the state of the art right now.
No, it was very helpful. Thank you. I think it's a hot topic of interest for some people to know, if a tracing experiment is needed to see if GAA is truly the precursor into neurons, or if creatine generated by other cell types might be the actual substrate for the neurons. So thank you.
Okay. Anyway, if I may add just one last consideration. GAA is certainly one way how neurons can make creatine. And moreover, if GAA can, as it seems it does, set up an alternative GAA phospho GAA system, this may be an interesting pathway to explore, disregarding whether or not neurons make creatine from GAA or by uptaking creatine itself. I hope I was clear.
Maurizio, this is Andreas Schulze from Toronto.
Hi, Andreas.
Can you hear me? Yeah.
Yes, very well. Hi.
I was slightly surprised to read and also hear today again that you are implying that guanidinoacetate is not epileptogenic. And you showed those experiments in your hippocampal networks. I wonder, did you use any positive control with using a different GABA A receptor agonist to prove that guanidinoacetate is not acting epileptogenic via GABA A receptor agonism?
Well, the experiment we did has been to pour, as you saw, we pour the guanidinoacetate into our slices, and then we are now, these weeks, pouring guanidinoacetate onto neuronal cultures. Now, under these conditions, what you normally see if the compound you pour onto slices or neuron is epileptogenic, like bikirulin maybe, for example, or penicillin, you see a clear alteration of the synaptic response. You see a clear alteration of the electrophysiology. If you pour bikirulin or penicillin onto these systems, you see an increase, a dramatic increase in population spike amplitude. You see a dramatic increase in mean firing rate of the neurons, and we did not see those effects. So we are quite confident to see that, at least based on what we saw so far, guanidinoacetic acid is not epileptogenic in the same way as, for example, bikirulin or penicillin or an MDA are. So this is what we can say and what our data show. Now, if it is epileptogenic in other ways, like Ferrada showed, it can cause oversprouting, that is possible, but it is not just the same as bikirulin or penicillin or known epileptogenics.
Thank you so much, Dr. Balestrino. We really appreciate all the wonderful speakers this morning, and thank you so much to everybody for your questions.
Can I ask you a question? Excuse me.
Oh, sure.
May I ask a question for Dr. Axerio?
Yes.
Yes.
Hi. Just very quickly, did you test only restoration of transport into malfunctioning transporters, or did you test or do you know of compounds that can increase transport in the normal transporter?
Both. I did both.
So you do have compounds that do increase transport in the normal creatine transporter?
Yeah. Yes.
Well, good question. And I look forward to hearing about that.
Great. Well, thank you so much again to our wonderful speakers and for all the questions. Time for a break? Or
Nope, we're moving ahead to the keynote.
Keynote speaker next. Thank you, everybody.
Thank you so much, Jenny. Appreciate the assistance, and thank you for spearheading the discussion here today.